Ladies and gentlemen, Assalamualaikum warahmatullahi wabarakatuh. At this conference, I will present the results of my research entitled Assessment of Genetic Variability on Sugarcane Seeds Propagated through Callus culture using RABD markers. As we know, sugarcane is an important crop as the main raw material producing sugar and energy source in the world. In Indonesia, sugarcane is one of the most important industrial crops due to sugar being regarded as a strategic food commodity. The slow supply of conventional sugarcane seeds often hinders the spread of the new superior varieties program, so tissue culture techniques is an option to accelerate that program. However, clonal variation was reported in sugarcane seeds from Callus culture, so this raises concerns about the use of sugarcane seeds from Callus culture. This study aims to determine the genetic variation in sugarcane seeds from cuttings and Callus culture using RABD and morphological markers. This study conducted in IcoLisT greenhouse and biotechnology laboratory. PS864 variety was as mother plant. Clonal variation were observed in all recurrence grown from its treatments, cuttings and Callus culture of 1 time and 3 times Callus subculture treatments, about 4 months. Using 42 morphological markers include parts of leaf, stems, and buds. Genetic variation was assessed using 3 RABD markers on 3 plants from its treatment, in which the first plants was selected as the recurrent that was most similar to their mother plant. The second and third plants were selected which showed clonal variation based on morphological markers. Table 1 shows the treatments of the sugarcane seed propagation methods and the number of sample analyzed for genetic variation using 3 RABD markers. This slide shows 42 morphological markers used to assess clonal variation in recurrence from propagation by cuttings and Callus culture with 1 time and 3 times of Callus subculture treatments. We can see table 2, the results of morphological screening on seedlings of PS864 at 4 months from cuttings showed distinct morphological variations in morphological characteristics of their mother plant. The most prominent differences were seen in root crack, auricle size, midrib detachment, and bud shape. Screening based on morphological markers on seedlings at 4 months from 1 time Callus subculture propagation indicated variations in some morphological characteristics that were not the same as the mother plant's morphological characteristics. The number of morphological traits that varied in the recurrent was also more than that of the recurrent from cuttings. The most varied morphological characteristics in the recurrent from 1 time Callus subculture were root crack, stem diameter, midrib detachment, hairy midrib, and auricle size. The number of seedlings that varied in these morphological characteristics were more than 50%. Based on morphological markers on seedlings at 4 months from 3 times Callus subcultures multiplication showed variations in 18 morphological characteristics. Very high variation of morphological characteristics in root cracks, stem diameter, auricle size, and hairy midrib. Meanwhile, the variation of other morphological characters in seedlings from 3 times Callus subculture was more varied than seedlings from 1 time subculture and cuttings, in the range of 23 to 46.15%, except for the growing ring and bud position of less than 20%. The results of the genetic similarity analysis based on morphological markers using NTC software can be seen that seedlings from cuttings were similar to their mother plant in the ring of 0.90 to 1. When observed based on the level of morphological similarity, seedlings from cuttings were more stable because their variation in morphological was lower than those from 1 time and 3 times Callus subculture treatments. This picture shows electrophoresis of PCR products using 3 REBD primers on seedlings from cuttings and 1 time and 3 times Callus subculture treatments. Figure 5 and table 5 show that all seedlings from cuttings have 100% of genetic similarity with their mother plant. In the population of seedlings from cuttings and Callus culture, there were differences in the value of genetic similarity ranging from 7% to 12%. Figure 6 shows that sugarcane seed propagation method affected the average percentage of DNA polymorphism in sugarcane lignan of PS864 variety. It can be seen that sugarcane seedlings propagated by cuttings have no polymorphisms. This indicated that the genetic stability of the plant is maintained at 100%. Meanwhile, sugarcane seedlings were propagated by Callus culture had a tendency to increase genetic variation by 7% from 1 time Callus subculture to 3 times Callus subculture treatment. This graph shows the average percentage of DNA polymorphism in seed skin from its propagation methods. The conclusion of this study are there was no genetic variation in sugarcane seedlings of PS864 at 4 months from cuttings, while in seed skin from 1 time and 3 times Callus subculture was a tendency for genetic variation of 4.6 to 11.6%. Somaclonal variations in seed skin from Callus culture were higher than that of seed skin from cuttings. Further research is needed to determine the somaclonal variation caused by Callus culture propagation in sugarcane until harvest, so that it can be known whether the genetic variation occurs in the genes for sugarcane yield trait. The results of this study indicate that the more Callus subculture treatments, the greater the tendency for genetic variation to occur in seed skin. Okay, this is my presentation, ladies and gentlemen. Thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh.